start. Thank you for joining the seventh and the final week of the stem cell training program. And this year, every week still a number of new trainees joining. So therefore, brief introduction. I'm Gitai Kim, director of the UCLA Human Stem Cell and the Genome Engineering Center. And our center focuses on both collaborative independent research, and then also we provide core service in IP cell reprogramming and the gene editing by the robotic automation led by Dr. Hyo-Kyung Cha and Dr. Dennis Arthur. You can access this service through the core-to-core partnership program through your institution, most likely through your animal core, stem cell culture core, cell culture core, organoid core, or existing stem cell core. So this program is supported by some California Institute of the Regenerative Medicine in collaboration with industrial partner, Thermo Fisher Scientific. And we appreciate a strong participation through this program and also continued encouragement from training across the multiple institutions. And in the final reminder, participants who attended at least 6 out of 7 lectures will receive the certificate of the training completion. And please ensure when you log in the Zoom and you log in the Zoom with your full name and for attendance tracking. And if you need, and then just write your full name and the email address in the chat box, the person who did not log in with your own account or that kind of things. And if you are attending without registration, even final minute, just look at the chat box, and then I posted multiple different linkers and get in there and register it. And you are welcome to ask a question at any time. So therefore, do not just hesitate and hold the question by yourself. Just turn on the mic and then speak up or post your question in the chat box. And then today, Tom will lead the lecture, and Oma and me and a number of the also leaders will answer the question in your chat box. Today, we will cover lecture 7, neuronal differentiation in 2D and 3D system. And we are pleased to have a special guest speaker, Dr. Christina Correa, and assistant professor of the pharmacology at Mayo Clinic, who will present on integrating system biology and artificial intelligence. And now I would like to hand it over to Dr. Thomas Forbes and also Dr. Omar Farah, followed by Dr. Correa.
Thanks, Kitai. Just get my slides up here. Can everybody see that okay?
Yep, all good, Tom.
And you can hear me okay?
Excellent.
Perfect. All right, I'll get cracking then. In the interest of time, we've got a lot of ground to cover today, and I think it is, call me a bit biased, but I think this is a very exciting topic as well. So as you see on the slide here in this pretty picture, the focus of our discussion today is going to be on neuronal differentiation, which I'll be giving. And then the second half of the lecture, as Kitai said, we'll be hearing from Dr. Christina Correa at the Mayo Clinic. And I believe she's got some really, really interesting stuff to talk about with regards to systems biology, integrating with AI, and how we use that to do things like advanced science. So should be a good one today. But for the first portion of the talk, really my goal, just to sort of set the stage a bit, is to talk a bit about some of the science and some of the science-based tools that we have at our disposal nowadays underlying basic neurobiology. And, I think neurobiology can be very complicated, although it doesn't have to be. So I think my goal is for this is to kind of take a top-down approach. I'm going to talk very briefly about sort of a parts list, if you will, of the nervous system. I'll introduce some of the bits and pieces then that make up, all kinds of things about, let's say, your experience of life, what you think about, how you feel, how you interact with the world around you, and the different parts of the brain that are responsible for those functions. We're also going to talk about, importantly, for the context of stem cells, how those different parts of the nervous system come to fruition, right, through the process of neuronal development. And then I'll segue into some specifics as to how we actually mimic aspects of that neurodevelopmental process in neuronal culture. And also touch on the anatomy of neural cell models. I'll give an overview of some of those workflow segments just from a high level, again, from our perspective at Thermo Fisher. After that, I'll dive into a bit deeper neuronal differentiation. I always think it's easiest to start this process and talk about this process first in 2D, and then I'll switch gears a little bit into 3D neuronal culture, and I'll give you obviously some nice examples and points of distinction for each. And then last, but not least, I always think this is interesting as well. I'm going to give you kind of a peek behind the curtains, if you will, and I'll talk about some of the things that we're doing internally at Thermo Fisher, and some of the trials and tribulations and data that have been generated by our neurobiology R&D team, who have been working on different models in 3D culture lately, especially one today with regards to dopaminergic neurons. So really big take home here is that my goal at the end of this is to give you a nice foundation about how your brain and your nervous system work from a high level, and then obviously sort of integrating into that where stem cells fit into the picture, and then leave you with a few useful resources at the end as well. So let's start by talking about the nervous system, right? And the reason I start with the nervous system, and not just the brain, is because the brain really is just one piece of this larger and much integrated network that we call the nervous system. Now, the nervous system obviously includes your brain and your spinal cord, which is called the central nervous system, which I've highlighted here in orange. But also all the connections between the brain, the spinal cord, and the organs of your body, for instance, which we call the peripheral nervous system, right? So all the parts of the nervous system, as the name implies, outside of the brain and the spinal cord, which I've highlighted here in yellow. Now, I'll be honest, at Thermo Fisher, we really don't do too much with the peripheral nervous system per se, but it's important to consider when we're talking about the brain, right? We really need to think about and consider the nervous system as a whole. And that's because even though the brain governs all other biological systems of the body, right, it absolutely by no means does so in isolation, right? In fact, as I'm sure you might imagine, it's readily influenced by all these other biological systems, for example, the peripheral nervous system and all its intricacies, right? And there's no real particular start or finish to this integrated and reciprocal system. That said, the brain is the central organ of the nervous system, really the belle of the ball, if you will. It's also one of the largest and I would say most complex organs in the human body. Now, what we do in neurobiology from a functional perspective is we consider the brain as kind of broken up into specialized areas. And under normal circumstances, what we hope will happen is that those specialized areas will work together in synchrony, right, to control the activities of the body, which I think this colorful image here on the left highlights quite nicely, right? So doing things like processing, integrating, coordinating all the information, let's say it receives from the sensory organs or the outside environment, and then making instructional decisions that are sent out in response. Now, the brain is also neuroanatomically divided into several distinct regions or lobes, right? We've got the frontal lobe up here, which is responsible for things like problem solving, judgment, motor function. The parietal lobes, right? Those manage things like sensation, body position. The temporal lobes, those are involved in things like memory and hearing. And then the occipital lobes, right? And those contain, for the most part, the brain's visual processing system. And so, as I said, and kind of alluded to, what happens collectively and throughout life as we develop is these different lobes integrate, right, to help us survive and then ultimately serve as our portal for making sense of the world. And as you see down here at the bottom left, in the very most basic manner, the brain does that through 5 different mechanisms: sensation, perception, feelings, thoughts, and behaviors, right? And again, at its core, the brain really is trying to do one thing: to take all those perceptions of the outside world, merge those with perceptions of the inside world, right? And then link them in a way that allows us to respond to our environment appropriately, right? And we do that through behavior and output. And as you might imagine, right, this process is very much a constant medley, if you will, of integration, or in some cases like disease, right, disintegration that can occur over the span of our human life, right? Now, functionality aside, if we were to dive one level deeper and consider the structure of the brain, it's comprised of the gray matter, that's mostly the neurons and unmyelinated axons, and what's called the white matter, and that's all the fatty tissue in between that's composed of what's called myelinated axons and blood vessels. And a really simple analogy that I was taught is to think of the gray matter as your computer, and the white matter as all the cables necessary for that computer to operate. Now, one level deeper, right, the brain's made up of billions of what's called nerve cells or neurons, right, and a supporting cast called glia, which we'll talk about in a moment, all of which collectively communicate through almost trillions of these tiny, tiny, tiny little connections called synapses. And so think of it as this vast network of cellular and electrical circuitry that you see here on the right, that allows the nervous system to function, as you see here on the left. Now, again, if we're talking about the nervous system, I do want to get a little bit more specific, right? Because it's not just this big loop of wires that connect the brain to the spinal cord, right? But rather billions of these tiny little nerve cells or neurons that comprise this vast network. And that's what a neuron is. It's just a nerve cell. And again, they're separated by these tiny little gaps called synapses. And those synapses are where the chemicals or neurotransmitters from one neuron are essentially spit out. And then the next nerve cell in the series detects those chemicals, and it passes electricity down its length to the next nerve cell, and so on and so forth. And so really a good way to think about your body and all these integrated nervous system functions that occur, right, minute to minute, day to day, right, is that you're just a flow of electricity between these different nerve cells. And Omar certainly heard me say this before, but I think we often overcomplicate our experience of the world, making it seem like there's like a movie playing inside us or something, right? But that's grossly, grossly overcomplicating it, right? In its very, very simplest form, the language of the nervous system, right, is really just electricity. Now, within the nervous system, there's a number of important cell types too, right? And so during this process of neurogenesis, as I put down here on the left, which is something that occurs throughout life, neural stem cells differentiate, right? And as they differentiate, as we talked about in the past, they become any one of a number of structurally and functionally specialized cell types, like neurons, as I said, the control cells, if you will. And these are the electrically excitable cells of the brain, the spinal cord, and the peripheral nervous system. And then, as I said, we've got cells called glia, which help support those neurons and help maintain their environment. And there's really two types of glia that I want to highlight for the sake of today. We've got oligodendrocytes at the bottom here, which make a nice substance called myelin. It's a fatty kind of white substance, right? And what that does is it wraps around the neuronal axons, so the elongated portion of the neuron. And when this wrapping occurs, what it does is it allows us to effectively communicate those cells with one another, right? Hence things like your reaction time and processing speed. We've also got cells called astrocytes. Astrocytes are just about the most abundant cell type in the brain. What they do is help provide physical and metabolic support to basically help keep those neurons happy and healthy. They also play a really important role in the brain's response to injury. One other cell type just to call out, I've not listed them here, but the hot topic at the moment, you may have heard people talking about them, are cell types called microglia in the brain. And as the name kind of implies, microglia are a type of glial cell, right, that you can think of as almost like the resident macrophage of the brain and the spinal cord, right? They absolutely interact quite closely with other cells like astrocytes, each of which influencing each other's functions, responses during things like health, disease, and development, right? But a big point of distinction here is their origin. So astrocytes, for example, they're derived from ectodermal neuronal progenitor cells, while microglia are derived from the yolk sac during embryonic development. And so again, big take home here is that it's this vast network of cells within the nervous system that allows for the rapid propagation of these electrical impulses. And these electrical impulses, right, or electrical activity at its core, is what allows us to respond to the environment appropriately. So where do these cells come from, though, right? This kind of dynamic electricity fueled functionality of the brain is something that begins way before we're even born, right, through the process of neurogenesis. And that process, neurogenesis, is the brain's ability to create new neurons and connections between those neurons. Now, neurogenesis, right, is obviously really, really important as an organism's developing, but it's also something that continues in certain brain regions after birth, and in fact, even throughout the lifespan. Obviously, a bit diminished as we get older, but keep in mind, you don't always need a lot of new neurons to really disturb the network or refine and tailor the nervous system. And so during neurogenesis, these neural stem cells differentiate, right? And they then, as I said, become any one of a number of structurally and functionally specialized neuronal cell types. And this obviously happens at specific times and regions in the brain. And we won't get too much into that here. But neurogenesis is especially important during development in the front part of the brain, right? What's called the prefrontal cortex. The prefrontal cortex is the last part of the brain to develop. Undergoes massive changes until, I'd say, about the mid-20s or so. It's also, importantly, the part of the brain that helps control things like actions, behavior, emotions, right? And if you kind of take one step back and think about that, that's a big part of why teenagers, for instance, often make quite impulsive decisions that they might find rewarding, right? Now, along those lines, when you find something rewarding, there's an area of the brain called the striatum. The striatum is very, very responsive to rewards, right? And it releases a chemical called dopamine. We'll talk about dopamine more later on, but dopamine is really, really important in things like learning, rewards, motivation, etc. And so the take home here is that the brain essentially develops from back to front, right? And again, by allowing for that constant refinement of those neuronal circuits that occurs throughout the life, it's that neurogenesis that helps contribute to structural and functional remodeling that, again, helps us optimize the cognitive response. So neurogenesis specifically refers to the generation of new neurons from neural stem cells. But again, if we take kind of one half step back here and consider neurodevelopment on the whole, this is something that very much encompasses the broader process of nervous system function, obviously including neurogenesis, but other crucial steps too. And neural development, I would say, really involves three key processes. First of all is proliferation, right? So think about that, increasing the number of cells. Once the neural tube is closed, neural proliferation begins to occur in an area called the ventral zone of the neural tube. And what's, I would say, quite interesting here is that newly formed neurons may remain where they are in that spot in the neural tube and continue to divide, or sometimes they migrate to other parts of the nervous system. So the second thing we have for a step is migration here, right? In other words, moving cells to their proper location. And in the nervous system, neural migration remains often within the confines of the neural tube, right? Whereas, say, for neurons of the peripheral nervous system, right, neural migration might take place across almost different neural regions altogether, right? One thing to keep in mind here that I think is very interesting is that migration tends to follow an almost inside-out pattern, right? So neurons travel from the inside of the neural tube, again, known as the ventricular zone, outwards towards their target location. And then finally, the third big step here is differentiation. This is a nice theme for us throughout this course. And so neural stem cell differentiation is really important because it takes us from those undifferentiated neural stem cells all the way through to specialized neurons. Obviously, as you might imagine, as I have on this slide here, differentiation is something that involves quite a few changes, right? Changes in things like gene expression, changes in morphology, changes in function, right? But again, all ultimately leading to the formation of those various neuronal subtypes, each of which has your unique characteristics. So in this case, neural stem cells, right? They're capable of self-renewal and differentiation. And again, they undergo morphological changes, including the development of the cell types that we talked about, dendrites, axons, oligodendrocytes, right? Which are important for communication. And as you see here on the slide, there's a lot of different signaling pathways involved, as well as transcription factors that help guide that differentiation process as those cells eventually acquire their specific functions, form those connections with other neurons through those synapses that we talked about. And again, as we grow older and older, we get better at things, refining all those complex neural network that ultimately drive superior brain function, right? And again, as I think neuronal differentiation is so important. It's such a big aspect of who we are, how we function, how we integrate and survive in society, right? And it obviously has big implications for things like brain health and disease. Okay, so we've set the stage a bit and we've got this dynamic, very electricity-fueled organ in the brain, right? And it sits right inside our heads. But it is right inside our heads, right? Underneath this rock-hard skull. So how on earth do we study it? Well, you may have seen this slide before, but the long and the short of it is we've got a few different model systems that we use to study the brain, right? Starting on the left here, and this list by means is no means all inclusive, right? There's loads of other types of models as well, but I've put what I think are the most more common ones here. Starting on the left, we've got planar.
Yeah.
Sorry, Omar, go ahead.
No, that was not me, Tom. That was somebody else.
Oh, okay.
I think they were just cut off. Yeah, you're all good.
Yeah, it's all good. All right, so starting on the left here, we've got planar or 2D culture models. These are readily available. Loads of folks use them. They're fairly cost effective. They're easy to manipulate. But if you think about planar or 2D models, right, they lack a lot of the very specific cellular architecture that we just talked about, as well as the cellular diversity that we need to closely mimic human disorders, right? And again, this is especially true in areas of the body like the brain. Neurobiologists often use behavioral studies, second one in here, and those are nowadays typically performed in rodents. Although, as we've talked about, we're starting to get away from those with the new approach methodologies or NAMs. But behavioral models are a nice bridge between the inner workings of an organ like the brain and functional output. But you've always got to remember they're by no means directly translational, right? Because we're always dealing with animals after all. And I would say keep in mind too that a lot of adult onset disorders, like neurodegenerative disorders like Parkinson's disease, which we'll talk about today, what they tend to do is impact our physiology long before behavioral changes are even detected. We also rely quite heavily on human tissue. Human tissue is great, right? It gives us a nice intact structure and therefore 3D microenvironment, but it obviously lacks the functionality component because we're typically not performing experiments in live tissue. Not to mention, there's always limited availability with the amount of things like post-mortem tissue that researchers can actually get their hands on. And then lastly on the right here, we've got 3D models. And as Omar talked about before, there's quite a few different types of 3D models, but 3D models in the neurobiology space are often derivable from human cells. They therefore more closely recapitulate things like cytoarchitecture and functionality. And so generally speaking, 3D culture replicates, I would say, in vivo conditions much more accurately than some of these 2D systems. And cells cultured in 3D give us data that are therefore more physiologically relevant, right? So closer to that of cells in their natural environment. Now, at Thermo, there's really two types of 3D models that we work with in the 3D space, and those are suspension culture and organoids. And we'll talk about each of those a little bit in the context of brain throughout this talk. But before we get into 3D, I wanted to start with, as I said, sort of laying a foundation in adherent culture. And kind of painting a picture, if you will, of what this might look like in a dish. And so this slide here is a nice, I think, high-level overview of the basic anatomy of 2D neuronal cell culture. Obviously, you'd like to be able to monitor, manipulate your culture conditions as you need to, keep the maintenance of healthy cells, right, giving you relevant and reproducible data. And so one of the things that we always first start with is the plasticware, right? And there's loads of different format options here, each of which offer hopefully somewhat of a controlled environment for growing neural stem cells by giving those cells a nice surface for attachment and expansion. Now, the thing is, though, neurons typically need a good amount of assistance in adhering to surfaces. And so we need a matrix to coat on top of that plate. And so matrices, as we've talked about, support things like attachment, spreading, neurite outgrowth of neural stem cells, and give them a more in vivo-like environment. And in Thermo, in the neurospace, we typically use either Geltrex, poly-D-lysine, or laminin. Laminin is probably, in my experience, the one that you'll encounter the most. And some folks will also pair poly-D-lysine with laminin for double coat, if you will. When we're choosing a matrix for neurobiology culture, there's obviously a lot of considerations, right? We have to think about things like surface chemistry, cost, other experimental parameters, right, for your specific setup. And then thirdly, we need cells, right? And there's, again, lots of flavors for cells, primary cells, iPSC-based neuronal culture, which I'll talk about in a moment. And then lastly, we need a cell culture system, right? Something to support the growth of these neurons, right? And this is another really, really important consideration, right? Our media choice, our media system, right? Because again, as I've mentioned in the past, not only does good media provide you with all the different nutrients, all the different growth factors, and really set the stage with the experimental conditions that you need for neuronal growth and differentiation, but it also really helps regulate things like pH or osmotic pressure of the culture, all of which, as you might imagine, are really, really important for maintaining the health and functionality of cultured neurons. And cultured neurons are amazing, but they are very, very, very finicky. They can be a real pain in the butt sometimes to work with. Anyway, for an example of media here, I've highlighted our B27 plus neuronal culture system, which we've got some really nice data with as far as enhancing neuronal survival, maturation, electrophysiological activity, and so on and so forth. Okay, so on the last slide, I briefly mentioned neuronal cells, right? And so kind of expanding on that, in the neurospace, most protocols nowadays are optimized with neural stem cells that are derived from either primary cells, as we see on the right here. So those are directly derived from tissue, or induced pluripotent stem cells, as we see on the left here, and we've talked about in previous lectures. These cells are reprogrammed, they're differentiated into specific cell types through a workflow that would look something like what you'd see here. Really another way to think of this then is to separate this neural workflow into two pillars. We've got the stem cell pillar on the left, the primary pillar on the right. I would say the primary pillar can be a bit more simpler, right? It involves isolation, separation of neurons from, say, fetal brain tissue. Sometimes customers choose to do this themselves. They want to isolate and separate their cells in the lab, or sometimes folks will purchase cryopreserved neurons as well. And again, they come in a variety of different species and subsets. On the left here, the stem cell pillar, as you might imagine, is a bit more involved, but allows you to really tailor and optimize that experience. Starting in the top in the gray box here, we begin, as we've talked about throughout the course, by reprogramming somatic cells into iPSCs, right? We then derive our neural stem cells, or NSCs. This is, I'd say, really the first step in producing actual neural cell types from pluripotent stem cells. Just as an example here, we have something called neural induction medium that we know can convert pluripotent stem cells into neural stem cells in about a week or so. And then at this point, there's neural stem cells, if we want, can be expanded or in many cases differentiated into specific subsets of neurons or glial cells for different downstream applications, as you can appreciate along the bottom here. I'd say nowadays probably it's about a 50-50 split, maybe a few more folks heading in the direction of the iPSC route, but there's certainly benefits to each system, and both are very, very functional and useful in the lab for generating good data. Again, in our case at Thermo, we, as I mentioned, can mimic that differentiation process using neural induction medium. This is a serum-free medium. It provides high-efficiency neural induction, as I said, in about a week or so, all without the need for an amyloid body step. We know that NSCs generated using this NIM or neural induction medium have high expression of important markers, SOX1, SOX2, nestin, and so on and so forth, and that they can be differentiated, as you see on the bottom here, into these pretty pictures of neurons, astrocytes, and oligodendrocytes. Okay, so that's 2D. Let's, now that we've set the stage, segue into 3D. Omar, any questions or anything in the chat that needs addressing?
So far, so good, Tom. We have one question about whether there's a need for specialized plasticware for neuronal culture.
In some cases, yes. Not always, as long as you can get this neuronal cells in 2D culture to adhere. In 3D culture, a little bit more, yes. I think plasticware becomes something that's, I don't want to say a more important consideration, but a more impactful consideration in 3D culture. And as I talked about, I'm going to sort of shed a bit of light or a peek behind the curtain, if you will, on some of the processes that our R&D team went into in 3D culture. And so I'll actually share with you a couple of different types of plastics and what we use and what gave us the best data in that space.
Yeah, and I would also say that in even in those transitional kind of midpoints, right around 2.5, right, D culture, if you will, things where you have co-culture applications potentially using neuronal cells with glial cells and seeing migration patterns, things like that. I've seen some of that research where you could use things like inserts or insert plates, right, that would require some sort of specialized plasticware. But yeah, I would agree, Tom. I think mostly on the 3D side is when we start to look at the plastic itself, aside from the coating that's on the plastic for traditional 2D culture.
Yep, exactly. Okay, let's segue into 3D here a bit. And again, we'll set the stage with suspension medium first. When we're modeling diseases, our pluripotent stem cells are, I would say, almost invariably differentiated into specific cell types, right? And we do that, as we've talked about in past lectures, with or without gene edits, again, to help us sort of recapitulate disease-relevant phenotypes. And so as far as differentiation products go, just to kick us off here, we have a number of products that align well with our suspension medium stem scale. And again, this is not a plug for stem scale whatsoever, but I think this is important because, as I said, now we can begin to focus on elucidating the 3D portion of differentiation, right? Allowing us to then make much better models that more closely recapitulate the human state. And so using our aforementioned neural induction medium, our NIM system, what we're showing here first is proof of concept that neural induction can be initiated directly from spheroids, so 3D culture in stem scale medium. What we did is we adapted our 2D monolayer protocol shown across the top here. This is comprised really of 3 stages, right? Induction in dark blue, expansion in light blue, and then maturation in kind of this purpley color here, different periods of these neuronal stem cells. What we see across the bottom half of this timeline now are protocols tested for spheroid differentiation and suspension, again using stem scale media here. What we did is at day 0, performed a simple media change into induction media. And after that, what the 3D protocol allowed was a good deal of flexibility. You can see that by the differences in the number of days here with regards to the first 2 steps of the protocol, induction, dark blue, and expansion in light blue. Importantly, though, and I would say quite impressively, on day 11, we're able to attain a similar, if not greater, number of neural stem cells to that seen in about 27 days of adherent culture, basically cutting those induction and expansion periods in half. And then by about day 32 here, we see the same, if not more, mature neurons as otherwise seen in adherent culture at about 48 days or so. So initiating neural induction from PSC suspension culture, if that's the route you choose to take, can allow for a shorter duration of induction and ultimately higher yield of mature neurons in considerably less time. We do have the capacity as well to scale this differentiation protocol across different kits. So I'll show you here what we observed with dopaminergic neurons. In this example here, we're using at the top right our PSC dopaminergic neuron differentiation kit. What this does is enables differentiation of PSCs into functional dopaminergic neurons. We adapted the kit to basically initiate expansion of spheroids, again, in stem scale medium, allowing for the production of the same amounts of these FOXA2, OTX2 co-expressed neurons in about 25 days in the 3D protocol versus 10 days more or 35 days in adherent culture. FOXA2, just to give you a bit of background, it's one of the first transcription factors expressed in the ventral midbrain. There's also some really interesting and cool papers out there on gain and loss of function experiments indicating how FOXA2 is really, really important and necessary to produce midbrain neurons during development, but that it's also expressed down the road as well in adult dopaminergic neurons where it helps regulate their survival. So FOXA2 is super, super important. OTX2, bit of background, it helps regulate the expression of proneural genes, things like MASH1, ASCL1, neurogenin2 for all you neuroaficionados, to help promote neural differentiation and, again, help proliferation of those midbrain progenitors. So both of these, without getting too much in the weeds, are really crucial transcription factors that we know play an important role in the development and maintenance of this specific subset of neurons, dopaminergic neurons. Okay, so that was spheroids, right? One type of 3D culture. But what about 3D models like organoids, right? And again, I want to just backpedal a tiny bit here and give kind of a quick brief side-by-side comparison of spheroids versus organoids. I'll be honest, I was absolutely confused by this in the past. They are similar in some regards, but they are also quite different in others. Spheroids and organoids both give you the opportunity to create complex 3-dimensional models, which, as I've said, these models more closely recreate in vivo-like tissue and organ conditions. But which model do you choose? Which model works worse? Well, it obviously depends on the type of application that you're working with. These spheroids are 3D cellular aggregates. They can be composed of one or more cell types. They do exhibit, as I said, enhanced physiological responses, again, especially over, say, 2D culture. Spheroids often retain the starting characteristics of the cells, but they don't spontaneously undergo differentiation or self-organization, whereas organoids do, right? As you see here on the right, these are 3D structures. These organoids derive from, say, PSCs, sometimes also from organ progenitors, so tissue-specific stem cells, in which the cells do spontaneously self-organize into differentiated functional cell types, right? And also which I would say more closely resemble their in vivo counterpart and therefore mimic at least some function of that particular organ. Now, structurally, spheroids are typically created, as Omar talked about in the past, in a scaffold-free environment. We do this by placing the cells in suspension colonies, typically without the aid of things like extracellular matrices or other physical support. Organoids, a bit trickier, admittedly. These are typically developed in media containing very specific organ-specific growth factors. They typically do require a scaffold, like an extracellular matrix. And again, I'll talk about the importance of that as well as the plastics when I show you some of the work that we've done on our end. But what that plastic and that extracellular matrix does, it helps encapsulate the cells, provide them then with an ideal growth environment, and it's that aids in their self-organization. Another thing that really helps with self-organization is polarity, right? That's a really important point of distinction as well between these two different types. Spheroids sometimes exhibit polarity, sometimes they don't, and that has a lot to do with them essentially lacking a defined architecture. While I would say by their very inherent nature and tissue-like architecture, organoids do typically possess some polarity, and this polarity or charge helps in their migratory and self-organizing capacity to become these mini organs, right? As the name implies, organoids. And then, as we discussed earlier, right, as with any model system, there's certainly advantages and disadvantages of each. I've just put a couple on here. Again, this is not an all-inclusive list. Spheroids are highly scalable. They're highly reproducible. They're great for things like high-throughput screens. Organoids are a bit more complex and in vivo-like, right, meaning they better resemble cell structures and that microenvironment, which can be really useful for things like targeted research, drug discovery, etc., very focused disease models. So let's kind of piggyback off of that and focus a bit more on neural organoids now. Again, I always like to kind of tell a nice storyline and talk about things from sort of a historical perspective. We've been studying the brain for ages. I would say not particularly well until recently. But again, keep in mind, as I talked about in the beginning, the brain is really, really notoriously difficult to study. Again, not only is it locked inside the hard part casing of our skull, where it absolutely hates to be disturbed, right? There's a famous neurosurgical saying where they say, once the air hits your brain, you're never the same, right? The brain doesn't like to be bothered. But also another thing to keep in mind here is all those neurons and all those connections between those neurons that we talked about, right? They're so small and they're so complex that, tracing and modeling those paths, right, and all the signaling dynamics involved, right, makes it really, really hard to investigate, not to mention the fact that we can't really even get in there. And so historically speaking, there's been kind of a hang-up, if you will, with a lack of appropriate in vivo models paired with that inaccessibility to human brain tissue that I would say until recently has very much hindered research in this space. And so neural organoids were a really exciting kind of new thing on this in the neurobiology scene because this allowed us to better evaluate things like neural networks, cell behavior, drug screening, disease modeling, right, all in a much more physiologically relevant manner. And they're, in a lot of ways, I would say, quite an answer to the problem because they give us good insight, at least, into things like basic developmental and even translational research. I will say, though, they're not perfect. As with a lot of techniques, there's a lot of diversity and structural complexity of brain organoids that can depend on things like how long you actually culture them, right? The cell types and cell specifics that you're using. Another thing to keep in mind with brain organoids is that even though we get to a mature state with them, so about 6 months, 4 to 6 months or so, organoids do possess important features, right? Progenitor zones, regional identity, right? Integrated circuitry, gene expression patterns that, yeah, they do very much mimic some of the spatiotemporal organization of the brain. But again, they're not perfect. There's certainly limitations, and I put a couple of those down at the bottom here. One of the biggest issues with brain organoids is that even at that long-term culture, at say 4 to 6 months of organoid development, they still, for the most part, only resemble about a second, maybe early third trimester fetal brain, right? And so they are useful tools, but a lot of the complex patterns that you might imagine then of neural activity that we would see in the adult brain can often be lacking in these organoid models. Just a couple of other limiting factors just to mention here for transparency are often that of vascularization, right? And so along those lines, organization, structure. With regards to vascularization, right, blood vessels play such an important role in structural integration, in nutrient delivery. And because a lot of the neural organoid models don't always contain functional vasculature, this can be a huge factor in limiting aspects of neuronal differentiation and maturation, which, as you might imagine, just don't occur without sufficient oxygen and nutrient supply. The last thing, not to pile on to neural organoids here, for as far as limitations go, is variability. There is a good amount of variability between organoids, between protocols, and this could sometimes amount to limited reproducibility. We see protocol to protocol, often substantial differences in morphology, size, cytoarchitecture within the specific organoid. Again, all of which I think we need to keep addressing to help advance physiological relevance of these specific organoid models. But as we always talk about, there's a lot of smart people out there doing some really, really cool work in this space. So I think we're only going to continue improving with these neural organoid models. Now, as far as the models go, there's been a number of approaches for generating neural organoids. From a high level, we broadly divide those into either unguided or guided protocols. As we see across the top here, brain organoids produced through unguided protocols, the so-called cerebral organoids. Those are spontaneously differentiated from pluripotent stem cells without the inclusion of extrinsic factors, right? And the seminal protocol for this was developed by a very famous researcher named Madeline Lancaster and her colleague. And this is actually, truth be told, really the foundation for our neural induction protocol too. What's really, really cool here with these whole brain or cerebral organoids is that within them, they contain various brain regions or regional identities, right? And what that means is we can almost collectively investigate whole brain interactions, right, with this unguided method, right, using one of the different tools that we have at our disposal. And there's a lot of different tools out there nowadays as well. I think, again, this is another space that's advancing so rapidly. You look at some of the pictures from neurobiology, some of the electrophysiological studies and data that folks are getting nowadays. It's absolutely amazing. Now, with regards to ePhys or electrophysiology, again, many researchers use and rely on these electrical features. Remember, we talked about electrical activity in the brain, right, as maturation criteria here for these brain organoids. Now, in contrast, though, to these unguided protocols at the top here, we've also got guided approaches. These are maybe a bit more of a modern technique, which do utilize extrinsic signaling factors to then almost choreograph differentiation, right, to produce region-specific brain organoids, almost allowing us then to pattern to these desired regional identities. And there's loads of different things that we can develop here and different signals at play, right? Sonic hedgehog, Wnt, fibroblast growth factor, just to give an example of ones that are important. And again, we won't go into detail here, but I think this figure highlights quite nicely how different guidance signals influence that patterning and morphogenesis throughout the brain. Now, last but not least on this slide, both of these approaches obviously have their advantages, as we see on the right here, especially with regards to diversity and consistency of neural organoids. Generally speaking, the unguided or self-pattern protocols are really good for developmental studies or studying neurodevelopmental disorders, where we want to look at communication between different brain regions. And again, that's because these unguided protocols, these cerebral organoids, display multiple regional identities all within a single organoid structure. Nowadays, though, a lot of the folks are going in the direction of guided protocols. We can control a lot more of the variables with this, cellular composition, reduce variability. And again, that's because the addition of these specific growth factors helps us determine cell fate a bit more, and therefore neural identity is a bit more reproducible. I will say, though, some of these small molecules that we need for patterning are not cheap. Again, not to mention that this can take quite a long time, sometimes on the order of 4 to 6 or even 8 months to develop some of these structures as well. There's loads we could unpack here. Again, super interesting topic, but there's lots to consider when deciding on suitable brain organoids and what you're actually looking for with regards to a specific study. I do want to touch a little bit on some of the things that we offer and we talk about at Thermo in our space. We've got some nice data in the 3D neural organoid space. What we're looking at here is an example of brain organoids produced through an unguided approach, right? So without the inclusion of those signaling factors. It follows an established workflow, much like you'd see across the top here. We typically start from PSC culture, aggregating cells in suspension to form what's called embryo bodies. Those cells in clusters are then capable of neural induction, neural patterning, and then finally growth and functional maturation into the whole brain or cerebral organoids. And again, we've got some really nice resources here in this space, and I'll include some of the links for those at the end of the talk. Just quickly along the bottom here, we're looking at data with regards to embryo body formation with or without Revitacell supplement here. Revitacell is something that we offer that allows us to improve cell viability and recovery and also reduce variability. Basically what it does is it minimizes the impact of stress on cells. But as you can see in these pictures at the bottom here, what we found was that the addition of Revitacell improved embryo body formation in a couple of different cell lines and a couple of different dissociation reagents. Okay, so that was the whole brain model, right? But we've also touched briefly on those organoid models that recapitulate different regional identities through the so-called guided approaches, right? Where again, we utilize extrinsic signaling factors to choreograph that region-specific pattern. So we're shifting now from sort of a whole brain organoid model to one that pertains specifically here to the forebrain. Again, that's that big area up here in the front. The forebrain is by all means the biggest region of the brain. It's super important. It does an awful lot of different things, receiving and processing sensory information. It helps us think, it helps produce and understand language. It gives, I would say, final say to a lot of our autonomic, endocrine, and motor function. So the forebrain is super, super important. Again, I won't go into too much detail on the protocol here, but what we show is that we're able to adapt 2D and 3D configurations to generate forebrain organoids in about a month or so, which is nice. It's quite a short amount of time. We also tested this protocol in a number of different media systems. We looked at enriched medium like StemFlex, suspension media like StemScale, defined medium like E8 or E8 Flex, again, to ultimately differentiate and form those organoids with that forebrain specific phenotype. Again, I won't go to the protocol too much, but PSCs are grown as aggregates in the media system of choice. We neuronally induce them about day 3. We expand them around day 8. And then again, depending on the format, we either dissociate the spheroids into individual cells, or we continue their development in suspension before introducing them to maturation media, depending on the format that you're choosing. Data-wise, what we did and what we're looking at here is confirming first that the organoids have a forebrain-specific identity. We're testing that here, looking at gene expression using a qPCR panel here. Just quickly, as far as the data goes, we're looking at quantified gene expression in spheroids specific to the forebrain, the midbrain, and the hindbrain. After induction, as you see here in dark blue, we start to see expression of forebrain markers like OTX2, which we talked about, being more pronounced in those forebrain-specific spheroids. The midbrain spheroids express FOXA2, which we talked about is super important, right? Again, it plays a crucial role in dopaminergic neurons. And then the hindbrain spheroids, as you see here, express more of those early hindbrain markers like LBX1. All 3 populations, as you see here, produced or express fairly similar levels of something called SOX1. SOX1 is a fairly non-specific marker of neuroprogenitors. And then down at the bottom here, we're looking at after a couple of weeks of maturation, a bit more qPCR data from forebrain, hindbrain, and midbrain. And again, without going into too much detail here, this data is in agreement with the literature as far as the gene expression patterns or levels that we'd expect in different regions of the brain at this maturational time point. Now, what we did next to try and establish a link or a convergence between gene and protein expression data is looked at immunocytochemistry. Here, what we did just quickly is looked at something called TBR1 expression. That's a reporter of a mature forebrain phenotype. We also looked at, as it's always important to do, non-neuronal genes as well, like GAL-C and S100 beta. These are really important in the context of glia. And we'd expect things like this in more complex models, right, that have different cell types and different compositions within them. Again, because again, that better mimics the natural environment. So just quickly, as we see on the right here, we're looking at forebrain cultures after about 2 weeks or so of maturation in either whole mount, section, or adherent culture. At the top here, we're looking at spheroids in whole mount. We're staining them here with TBR1, which again is specific to the forebrain, as well as HUC and D, which are antibodies that bind specifically to antigens present on neuronal cells. Below that, we're looking at images of 3D sections on the left and then 2D sections on the right. And again, using antibodies for different forebrain-specific markers and also non-neuronal markers. In each format, what we found is that colocalization here was indicative of a cell population, very much indicative of later stage forebrain. And again, this is in agreement, importantly, with the gene expression profiles that we saw here on the left. We then tested a method that allows us to look at using this protocol in leaner systems like E8, right, essential 8 media, allowing us then to kind of transition from defined medium to more enriched media system or so on and so forth to help develop these forebrain-specific organoids. We basically noted that the transition from defined to enriched media system was very, very much possible with this protocol and was actually shown in some cases to enhance spheroid growth and efficiency, which was nice. So there's flexibility to this protocol, which is always nice. What we did next, we wanted to improve neuron yield, first in a 2D format. So what we did is we dissociated spheroids into individual cells. We replated them for a couple of different passages in expansion medium, which we're showing across the top here on the left. These are spheroids cultured in stem scale suspension medium, with these colorful images across the bottom here showing expression of different forebrain markers, OTX2 and PAX6 specifically, after a few passages in expansion medium. Again, on the right, what we're looking at is qPCR data for gene expression profiles, this time in 2D and 3D culture after a couple of weeks of maturation, as well as for a number of different non-neuronal markers too, like GALC and S100 beta. Again, this really just, I think, highlights the complexity of these models, drives home their very realistic nature, and demonstrating that we can, in fact, make these models with flexible protocols and high neuronal yield, which is nice. This is, as I said, in a really important region of the brain. So it's good that we have a very robust model from which to study. We then looked at electrophysiological activity. Remember, electricity is really, really important in the brain. It's the fundamental currency of the brain. So we use ePhys, or electrophysiology, to help us map different spatial and temporal fluctuations in neural activity, really as a means to probe at the functionality of a given neural network. So again, I won't go into too much detail here, but what we're looking at is forebrain cultures capable of exhibiting electrical activity in 2D. As we look across the bottom here, we're looking at calcium oscillations in cultures after transfection with something called a synapsin promoter. We use that synapsin promoter to help drive expression of our calcium indicator. What we're looking at on the left is a spontaneous baseline activity in our dissociated cells. In the middle here, we see elevated activity after treatment with something called PTX, or picotoxin. It's a neuronal stimulant. And then lastly, on the right here, we can actually reduce activity in this model with a neuronal suppressant, in this case called TTX, or tetrodotoxin. These trace images here on the bottom basically show the intensity of fluorescence due to calcium channel activity over time, right? So functionality in the brain. And these little asterisks indicate the time at which each image was captured. Again, all shown really as a measure of spontaneous and chemically altered neuronal firing in 2D culture. Now, in 3D, we use an electrode array system in this case, which really is a nice way to study the brain and test the ability of, in this case, these differentiated forebrains model to fire action potentials. And action potentials is a term that gets thrown around quite a lot. You've probably heard of it. These are electrical impulses that send signals in the brain through like shifts or depolarizations in neural membrane potential. So what we did here in this case is we used multi-electrode array seeded with 3D organoids, and then we took action potential recordings over time. Again, I won't go into the nitty-gritty here, but neuronal activity and functional connectivity were detected. We know that neural synchrony increased, became more complex over time. And again, as I mentioned in the beginning, when we get synchrony and complexity of these neural networks, this is such a big and important component of integration that occurs neurodevelopmentally, right? And it shows signs then of functional maturation, right? Again, all indicative of a physiologically relevant model, much like I would say what we would expect to see within the human brain itself. And then last but not least here, again, I won't go into too much detail, but we wanted to generate a very specific population of these forebrain spheroids, a ventral specific population, so the lower front part of the forebrain, again, just showing us that we have some flexibility with this model. Again, it's nice to have these very specific models of regions of the brain, in this case, specific models that allow us to even study different interactions between those different brain regions, or even in some cases, the same brain region, as we're seeing here with the forebrain. So again, I won't go into too much on the data here for the interest of time, but I think this is a really good and very impressive model. Okay, so that was a guided approach, right, of forebrain organoids. And what I wanted to end on today is another example of the guided approach for generating neural organoids. In this case, though, midbrain specific organoids, which are really, really useful models to study Parkinson's disease. And we talked about Parkinson's disease a tiny bit in our introductory lecture. We talked about the importance of something called LRRK2, LRRK2, if you remember. LRRK2 is a widely expressed protein in the brain, but we know it's linked to a lot of the different pathogenic mechanisms, especially underlying things like Parkinson's disease. Again, Parkinson's is a very common neurodegenerative disorder. First and foremost, it's characterized by a host of motor symptoms, as we see here in this character on the left. These motor symptoms begin quite gradually, but almost invariably they get worse over time. You sort of pile on, if you will, with those motor symptoms to include disturbances in behavior, emotion, sleep, memory, and so on and so forth. And what happens here is neurons in an area of the brain called the substantia nigra that we know is responsible for reward and movement. They basically become impaired and die. And again, these are also the neurons that produce dopamine, which I've talked about, plays a massive role in how we do things like think, plan, or feel, right? People with Parkinson's also lose the nerve endings that produce norepinephrine. Norepinephrine is also really important in the brain. It helps us mobilize for action, right? Not only that, it helps dictate how we react to things like stress and anxiety. And so when you think of Parkinson's, think of the loss of dopaminergic neurons in this really, really important region of the brain, paired with almost a blunting of those different nerve endings, and that just throwing absolutely everything else out of whack here, right? As you see on the bottom right. So if we refer back then to those basic functions of the brain, sensation, perception, feelings, thoughts, and behaviors, you can imagine how loss of these very specific dopaminergic neurons and that blunting of the nerve endings could be very catastrophic. And it really is a massive problem, as you see across the bottom left here. And so, as I promised, just to shed a bit of light on some of the work that we've been doing in this space, what we're looking at here is experimental conditions, comparing a 2D and 3D floor-plate derivation, again using our PSC dopaminergic neuron kit. Just as a point of reference, as I've thrown this term around a lot, the midbrain floorplate is something or a region of the brain that plays a really important role in the development of dopaminergic neurons. The midbrain floorplate really acts as kind of a signaling center, if you will, that provides a lot of different cues for specification and differentiation of these specific cell types. So here we're looking at the top workflow showing 2D floorplate specification of culture grown in essential 8 medium. We know that this kit specifies midbrain floorplate in 2D, after which the progenitors are expanded through a few different passages. We seed those onto poly-D-lysine and laminin, as I talked about before, to produce those dopaminergic neurons by about day 35. In the bottom workflow here, we're looking at 3D midbrain organoid formation. We begin with PSCs grown in suspension culture, again in our stem scale medium. We first dissociate those PSCs. We then reseed them into suspension culture for, again, 3D floorplate specification. We then transfer those floorplate spheroids into an expansion medium, and then finally a dopaminergic neuron maturation medium, as you see here in these different colors, without further passaging. As I said before, compared to 2D cultures, specification and expansion can actually be shortened a bit for 3D cultures, which is nice. Again, all with equivalent expression of these different neuronal markers for floorplate, dopaminergic neurons, and so on and so forth. And so what I thought I'd do next is show you how we tested varied culture conditions to help us optimize this 3D workflow in organoids. Which, I think is important. It's important to make sure that you're using and refining the protocol that you're working with, right? There's probably, as we get further and further along in science, especially in an area of science as finicky and as tricky as neurobiology, you want to always keep looking for ways to optimize your process. So in this case, we're testing multiple methods to help initiate organoids. First and foremost, we looked at self-assembly versus aggregation of a defined number of our embryo bodies, right? In other words, in this case, static culture versus rotating culture, right? And then we also looked at naked culture versus extracellular matrix encapsulated culture, right? And in this case, these cells are naked because we did not add extracellular matrix or ECM. So again, just to give you a glimpse at this, for starters, what we found is that in these rotating plates, the embryo bodies tended to self-assemble. We've got quite a lot of them per well. What we found, though, in static culture was that the PSCs were almost forced to aggregate into a single embryo body per well. And again, talking about plastics, the wells that we used here very specifically were our low attachment Nunc-Sphera 96 well U-bottom plates, which were shown to produce these really nice cultures. And I'll show you a bit more on that in a moment. And basically, the U-bottom shape of these allows the cells to form those organoids without them attaching to the plastic, right? Without them sticking to the bottom of the plate. Now, again, data-wise, in rotating culture, what we did is we started from small spheroids on the top left here, about 2 days post-passaging. We then specified this, which led to a bit of epithelial outgrowth by about day 2. You then start to see a general increase in size, I would say. By about 3 weeks or so, as you see here, we start to get these kind of irregular, kind of funky-looking organoids. You see some blebbing here, kind of an uneven shape of these structures. I'd say quite a lot of heterogeneity, which isn't always a good thing. But as I mentioned on the last slide, when we seeded those dissociated cells into those U-bottom microwell plates, that led to aggregation. And what we typically see with that aggregation is in less than 24 hours, right? That happens even prior to starting specification. So we're already sort of ahead of the curve here with regards to rotating culture. So as you see on the right, we get these larger and I'd say more uniform spheroids even earlier on. They do admittedly become a bit irregular as that kind of epithelia expand around the perimeter. And so what we did next, as I said, is introduce extracellular matrix, or ECM, to these two different recipes. And what's interesting here is how we first tested the addition of this ECM. We did this either by encapsulating the free spheroids in Geltrex ECM at day 2 during floor plate specification, or we tried also adding a dilute solution of 2% Geltrex to the microwell plate at day 2. We couldn't flip-flop back and forth. What we found here is that you couldn't cross over, if you will, with and how you added the ECM in this case. When you tried to encapsulate in the U-bottom wells, it just became too hard to actually get them out of the cell themselves. And we couldn't use dilute ECM in the shaking or rotating culture. It basically just triggered massive aggregation, which is no good either. Interestingly, though, ECM encapsulation plays a huge role. And this is something that's out there in all the literature, I would say, nowadays. What we find when we encapsulate these cells in extracellular matrix is a big increase in epithelial outgrowth, as you see here by about day 5 or so in rotating culture. This kind of produces these, again, I'd say good size. They're large, but they're very irregular looking organoids by about 3 weeks or so. They've got less blebbing than before, which you can see also here, kind of this encapsulation around day 26 or so, being quite visible around the outside. In static culture, though, we saw even less blebbing, which is good. And I would say much more robust looking spheroids that have kind of a healthy amount of cavitation, which is a nice indicator of health. When we paired that with the dilute 2% Geltrex in these static microwells, we saw maybe a bit more modest, but still quite nice epithelial outgrowth, as we see on the right here by about day 10. But I would say the size and the shape of these organoids was much more uniform than in any of the other condition. These are really nice looking cells. And again, this is totally in agreement with the literature here, which suggests that kind of the concept being some rigidity within the media goes quite a long way to help improve differentiation and maturation. When we add Geltrex, in this case, at a low percentage, it doesn't solidify the media in the well. What you kind of create is this dilute mixture that has some rigidity to it, right? What that does is you can kind of make around this structure here. It helps keep those structures intact, right? Which is good as they're developing. So this is organoids, right? But what do the dopaminergic neurons look like using these different methods? And so again, after optimizing for different conditions, right? Self-assembly versus aggregation, naked versus encapsulated, so 4 experimental conditions, right? We looked at these midbrain organoids forming spontaneously in rotation on the left here, forming in rotation for 5 days prior to being encapsulated in Geltrex, assembling via aggregation in the 96-well U-bottom plates, or assembling in the U-bottom plates with 2% Geltrex, as we see here on the right. Now, 3 weeks after floor plate specification, what we did is we immunolabeled these cells, these organoids. We did this in whole mount first for a couple of different important markers here. I'd say the main one being TH, or tyrosine hydroxylase. Tyrosine hydroxylase is the rate-limiting enzyme in dopamine synthesis. So as you can imagine, it's very important here. We also looked at MAP2, or microtubule-associated protein, in red. We know MAP2 is a useful marker of neuronal differentiation. And then DAPI, which everyone uses in blue. DAPI stains DNA. It's a useful nuclear counter stain. So pretty ubiquitous in any culture setting. What we did is image these organoids in confocal on a high content platform here called the Cell Insight CX7. And we show this here as either maximum intensity Z projections across the top or a single optical plane as we're looking across the bottom here. Again, what we found is that ECM encapsulation drove the outgrowth of that epithelia, kind of resulted in a highly folded structure, if you will, with quite a few neurons. We see aggregation of PSCs in those U-bottom plates, again, with increasing complexity as well as increasing neuronal yield, which is good, especially when 2% Geltrex was added to the specification medium. And again, that was about day 2 to 5 of the protocol. Just quickly, you can see in the bottom left here that these free rotating organoids without encapsulation I'd say have a fairly simple structure. You've kind of got those MAP2 positive neurons ringing around the outside. We look further over, we see the organoids formed in the microwell have, I'd say, more TH positive neurons. You can start to see extensive neurites kind of burrowing in. Encapsulating that in that free organoid in ECM had, I would say, a similar effect. We've got a nice convoluted layer here of cell bodies. We've got more abundant TH positive neurons. But if we go over here all the way on the right, we've got this static organoid exposed to dilute Geltrex. This is a nice compact round shape. We've got a nice thick layer of TH positive neurons. And I think you can appreciate that very much in the top and the bottom images here on the right. So the big take home here is that ECM encapsulation in the static culture, in these UL microplates, allowed us to increase organoid complexity and dopaminergic neuron yield as well. Now, we're talking about the brain, right? And when we're talking about the brain, especially, call me biased, always talking about functionality, right? And how physiologically relevant things are. So in this case, we're looking at a substance called neuromelanin. And neuromelanin it's kind of this like reddish brownish pigment that we know is synthesized from L-DOPA, dopamine, right? It's commonly seen in a subset of neurons called catecholaminergic neurons in the brain, right? In Parkinson's disease, what we see is selective degeneration of brain cells that contain this dark brown pigment, this neuromelanin, especially dopamine-producing cells, as we talked about in the substantia nigra, shown here on the left. And so again, kind of piggybacking off of what I talked about before, what our team found was that the addition of extracellular matrix here really quite readily influenced the amount of melanin in these midbrain organoids. We show spheroids formed without ECM, or so-called naked organoids, right in the middle here. They had very little pigment. The rotating Geltrex encapsulated organoids on the left here, I'd say had more pigment, it's quite visible. But the organoids formed in these static microwells with dilute Geltrex, I would say you can see had the most neuromelanin. It was also visible earlier, which is nice, by about 5 weeks or so. And again, this more closely recapitulates what we'd expect to see in vivo. And so the big take home here is that progressive degeneration of these catecholaminergic neurons produces or causes a significant reduction in dopamine, right? And when we lose that dopamine, we start to lose those neurons. What happens then is we start to see the motor issues that we'd expect to see in a patient, right? And then when those cells, these dopaminergic neuron cells, are oxidized, we start to see neuromelanin, right? And we think that neuromelanin, we're not exactly sure how it happens, heavily contributes to that neuronal death, right? So it's sort of a vicious cycle, if you will, and actually worsens patient symptoms in those living with the disease. So all intricately aligned here, and all sort of big indicators of how pathophysiologically relevant all of this is in the greater context of what you actually see or expect to see in a patient with the disease. Now, electricity, right? I keep coming back to this recurring theme. It's the fundamental currency of the brain. So we need to, if we're looking at physiological relevance, evaluate electrical activity. In this case, spontaneous electrical activity, again, using MEA or multi-electrode array. What we did here is we plated 21-day-old midbrain organoids in MEA and multi-electrode array at 1 per well. We saw some spontaneous action potentials within about a week, which is nice. And that those action potentials are indicated by these tiny little black bars here in what's called raster plots. By about day 35 or so, we actually start to see, as these networks develop and refine, coordination of action potentials, even across multiple electrodes, which is super cool. These vertical pink bars here basically mark bursts of activity seen across at least 4 different electrodes at a particular point in time. What's really cool here, and this is one of my favorite things about neuronal studies, you can actually shut that off. We can silence that coordination if we want, in this case by adding or flooding the system with excess dopamine in the middle here. What that does is it suppresses dopaminergic activity. Without going too much into the weeds here, this is a subset of receptors called D2 autoreceptors linked to inhibitory G proteins that we know play a really big role in regulating and monitoring the activity of dopaminergic neurons. What's really cool, though, is we can actually have that coordination reappear following washout of that dopamine. Now, at the bottom here, after about 7 weeks or so 50 days or so, we see even stronger coordination of that activity. These organoids are still quite sensitive to exogenous dopamine, as you see here in the middle. But even after washout, the organoids still form, I would say, quite a robust network of active dopaminergic neurons, which, again, I think is just super cool. And then last but not least here, we're looking at multi-electrode array analysis with regards to functional activity and network formation in 2D versus 3D. You can see here at the top, 3D, as we've talked about, offers, if you want, a shorter protocol for specification and expansion, as well as I would say, you can appreciate in this graph here, more robust and synchronous activity in these plots called raster plots. We quantified this down here at the bottom. We know specification and expansion in the blue lines, as you see in the graph, gives a nice boost to neuronal maturation over 2D, which is represented by the gray lines. Just quickly starting on the left here across the bottom, we see higher activity or mean firing rates, greater network formation or synchrony, as well as more activity over time and over a wider area in the 3D protocol, in a couple of different cell lines as well, which is important. All of which I think speaks to the importance of 3D culture. And then last but not least, to make this a bit more of a complete story, this is a bit of data and how we implemented this in another context of disease modeling. We've talked about the different mutations in things like LRRK2 or SNCA, which we know are associated with things like familial or sporadic Parkinson's disease. And so what we're able to do nowadays, which is really, really cool, is we can look at the effects of these different mutations in isolation and derive different Parkinson's associated lines through gene editing, right? Using tools like CRISPR-Cas9, as we've talked about earlier on in this course, allowing us to create lines, as you see here in the bottom, that are heterozygous, homozygous, or even in many cases unchanged for the mutation, depending on the different SNPs that a researcher might want, which we can then culture all of those different lines to start differentiation. And again, all of this is now possible in 3D using these midbrain organoid models. As an example of that, we looked at these mutants exposed to something called rotenone here. These are alpha-synuclein or SNCA mutants exposed to rotenone. Rotenone is something that inhibits mitochondrial complex 1 of the electron transport chain that then boosts or increases oxidative stress, which is obviously not a good thing. And so what we did here, in other words, is evaluated how responsive the mutants were compared to the wild types to different stress signals in apoptosis. On the left, we're looking at Z-stacked images of a 3-week organoid stained with a dye called cell event 37. What that does is identifies apoptotic cell populations. We also looked at TH or tyrosine hydroxylase, and you can see by about 3 weeks or so, I would say pretty extensive damage to those TH positive neurites with increasing doses of rotenone. On the right here, we're looking at cryosections of 5-week organoids also exposed to rotenone. These are stained this time with an antibody called caspase-3. It's another useful marker of apoptosis, as well as tyrosine hydroxylase. And basically the take home here is that overall the level of caspase-3 indicates increased stress in the mutant organoids. We know that the mutants are more sensitive to rotenone, our stress signal, even at admittedly quite low doses, which again is in agreement with the literature, where we know that Parkinson's dopaminergic neurons are much, much more sensitive overall to any and all types of neurotoxins. And again, this mimics very much what you'd expect to see in a patient with the disease. All right, I'm going to wrap things up here just in the interest of time. I know this can be kind of a heavy topic. Hopefully we've diluted it a bit and kept it interesting today. I talked on a few different principles of basic neurobiology, especially with regards to the structure and function of the brain and how intimately those two things are tied to electrical activity, right? Electricity, fundamental currency of the brain. We also talked a bit about neural development in the context of neurogenesis and neural stem cell differentiation and how we're able to model these different features using 2D or 3D culture, right? And again, I shared a couple of examples of those today with regards to things like the forebrain, with regards to the midbrain, as well as gave you a quick glimpse as to some of the work that we've been doing in this space to mimic, I would say, really important aspects of health and disease. As always, I'll share these slides afterwards, and I'll put this slide on here as well, just as a quick overview or point of reference of what we've covered today, and I'll include some useful resources down here at the bottom. But I am going to stop there, as I said, in the interest of time. We have another really exciting portion of the lecture coming up. If there are any questions, feel free to pop them in the chat. Otherwise, Kitai, if you would like to introduce our next speaker, that would be amazing.
Yeah, thank you very much. Anyone just briefly 1 or 2 questions? And if not, just feel free to leave your question in the chat box. And Tom and Omar will continue to answer your question. And the next is